This is an overview of an article titled "The Genetic Control of Left Atrial Gene Expression Yields Insights into the Genetic Susceptibility for Atrial Fibrillation," published in the March 2018 issue of Circulation, Genomic and Precision Medicine. Atrial fibrillation is the most common arrhythmia in the general population and has a strong heritable component. Genome-wide association studies, or GWASs, have identified at least 23 loci associated with atrial fibrillation. However, for almost all of these loci, the causal genetic variants and the mechanisms underlying these associations are unknown. The authors propose that some of the loci might influence susceptibility to atrial fibrillation by altering the expression of nearby genes. These gene expression changes may alter the cell physiology and anatomy and, over a lifetime, predispose to atrial fibrillation. In the paper under discussion, the authors sought to generate a genome-wide catalog of genetic variation and gene expression in the human left atrial appendage in order to identify potential functional variants for atrial fibrillation. Tissue samples were collected from 265 patients who underwent elective cardiac surgery for atrial fibrillation, valve disease, or other cardiac disorders, including 235 patients of European descent and 30 patients of African descent. First, the authors used RNA sequencing, or RNA-seq, to generate individual gene expression profiles of 24,000 protein-coding genes and long non-coding RNAs that are robustly expressed in left atrial appendage tissue. Second, they performed a genome-wide genotyping using single nucleotide polymorphism, or SNP, microarrays, and for each gene, tested associations with variants located within 250 kilobases of the gene transcription start site. From this, they identified nearly 16,000 genome-wide significant expression quantitative trait loci, or eQTLs, in European Americans, and more than 700 eQTLs in African Americans. They were able to show strong cross-tissue replication between their eQTLs and eQTLs found in related tissues from the Gene Tissue Expression, or GTEx, database. Lastly, they performed allelic expression imbalance analyses as an independent method to identify genetic variants associated with gene expression. Even though only a small number of eQTLs were identified in African American patients due to the relatively small sample size, the effect sizes and directions of association for the top-ranked eQTL SNPs in African Americans were strongly correlated with the top eQTL SNPs for each gene in European Americans. The authors have made all of the significant SNP gene pair eQTL associations in European American patients publicly available through a searchable web application. The web address is shown here. The web application also displays the eQTL association values for the African American cohort. The authors then focused on the 23 independent atrial fibrillation risk loci identified by GWASs. Of these, 12 atrial fibrillation risk loci had lead SNPs with one or more genome-wide significant eQTLs, as highlighted on this slide. Notably, the top eQTL gene in each GWAS locus was not always the nominally attributed closest candidate gene. As an example, the top-ranked atrial fibrillation eQTL for GWAS SNP RS108-24026 on chromosome 10q22 is shown here. The most significant eQTL association for RS108-24026 was with the MYOZ1 gene, where the major allele was associated with lower expression levels of this gene. The second strongest eQTL association for RS108-24026 was with the gene located closest to the risk locus, SYNPO2L, which went in the opposite direction, with the major allele associated with higher gene expression levels. These findings suggest that altered expression of either or both of these genes might contribute to susceptibility to atrial fibrillation. Furthermore, the authors observed several other variants that were in linkage disequilibrium with RS108-24026 that had even stronger eQTL effects, raising the possibility that the GWAS lead SNP might not be the causal variant at this locus. Lastly, the authors examined the effects of genetic variation on gene expression using allelic expression imbalance, or AEI. This method uses variants that occur within exons, called indicator SNPs, and counts the number of RNA-seq reads containing each of the alleles in heterozygous individuals to give a measure of the allelic expression balance. Combining European and African American patients, the authors examined all genes for which there were at least 20 individuals who were heterozygous for one or more indicator SNPs, 5,153 genes in total. For each of these genes, the authors tested associations between allelic expression imbalance and each SNP within 250 kilobases of the gene transcription start site. They identified an AEI association for 1,248 genes, approximately a quarter of the genes tested. In many cases, the top AEI SNP differed from the top eQTL SNP, as shown in this example, for the SYNE2 atrial fibrillation locus. The top panel illustrates the expression ratio of the minor to major allele counts in each patient who is heterozygous for the indicator SNP, RS356482226. The colors of the bars indicate whether the patients are homozygous, in green, or heterozygous, in orange, for the top-ranked allelic expression imbalance SNP, RS2738413, showing clustering of homozygotes and heterozygotes. This variant was in strong linkage disequilibrium with the SYNE2 locus atrial fibrillation GWAS lead SNP, RS1152591. The second and third panels show Manhattan plots for the allelic expression imbalance SNPs and eQTL SNPs in this locus, respectively. These show excellent concordance, although a different variant that was in strong linkage disequilibrium with RS1152591 had the strongest eQTL. From this, the authors concluded that AEI analysis might pinpoint functional variants, as the AEI method controls for expression within each subject and eliminates much of the variability in the eQTL analysis. In conclusion, the authors have identified a short list of candidate functional variants that might contribute to susceptibility to atrial fibrillation. This study provides the first left atrial appendage specific eQTL browser for genetic studies of atrial fibrillation. This resource could facilitate the identification of functional variants not only for atrial fibrillation, but also for other cardiovascular traits and diseases.